Our next speaker is Radha. Just a second. Radha is doing her PhD at the University of Kaiserslautern, working in a number of areas such as visual search, analytic, holistic processing, eye movements, cognitive load, and handwriting research. Her primary area of interest are eye movements, scan pad, and working memory. Today, she will present her PhD work, which is recently published with Professor Cees van Leeuwenhoek and Dr. Andrei Nikolaev at the Laboratory of Perceptual Dynamics, Brain and Cognitive Unit in Leuven. The link will be posted shortly in the chat. And stage is yours, Radha. We can't hear you. Please unmute yourself, or I can unmute.
Yeah, I'm trying to.
Thank you.
Oh, all right. Thank you. Thank you for this opportunity to Neuromatch as well. And thank you for the organization. Today I'm going to be presenting some work that I did during my PhD at the Perceptual Dynamics Laboratory with Professor Case and Dr. Andre. So this work is part of a larger effort to study EEG during working memory tasks under free viewing conditions. So why free viewing? Why is free viewing important or different from traditional working memory tasks? Oh. I'm not able to control this, yes. Because eye movements, whenever there's free viewing and we have unrestricted vision, we tend to make eye movements and a lot of saccades. And what this brings is a lot of additional complexity, which are outside the purview of the experimental conditions that we are interested in. So as Anne just presented, what we know from quite some time is that when a saccade is planned, attention shifts to the subsequent fixation location before the saccade is executed. And this attention shift, turns out, is affected by a number of top-down processes, such as working memory, prior knowledge, and other task-related conditions. Another thing that happens when you talk about free viewing is this, refixations, which is essentially moving our eyes to a previously fixated location or area of interest. Now, this behavior is also common, and it has been observed in a number of different tasks, such as reading, visual search, scene viewing, pattern copying, and so on. And so this is a question that we were interested in. How does task-related refixation behavior affect saccade planning? And the highlight is that we were trying to do this under free viewing conditions. So to test this, this is the display we used, which is a visual search display, and with target T's and distractor L's. And as I told you before, what we are primarily interested in was a working memory task. So we had 3, 4, or 5 target T's among distractor L's, and this was our experiment setup. So participants were asked to look for these targets during the 10-second search. And what they had to do was remember the orientations of the targets, the target T's, in order to be able to do the subsequent task, which was a change detection test. And what happened in the change detection was that one of the targets could have changed the orientation. So they had to remember all of them to be able to do the task. And here is a sample display of the eye movements that we observed during our tasks. So as you see, yes, eye movements. And also sorry, what we found from some...
We couldn't hear you in the last 20 seconds.
Oh, sorry, I will just repeat that. So that we observed quite a few refixations in our eye movement data. And what we had also found from earlier studies is that there is a particular effect that the task relevance of an item has on eye movement behavior. So for example, it affects fixation durations. Fixation durations on targets are longer than distractors, which is kind of expected and understandable. But what we also found was an effect on refixation behavior, that more target refixations were made in correct trials and incorrect trials, and the opposite was found for distractor refixations. So what we thought was that target and distractor refixations are made for different reasons and are driven differently. Target refixations are made for rehearsal. which means that you have to go back and remember the orientation of the target and also know where the target is. Whereas distractor refixations are probably made because you've forgotten, and in the process of new search, all you have to remember is the distractor location so that you know you do not go back, which means that there is probably a difference in processing load or working memory load between targets and distractors. And this is what we expected to affect Saccade planning. So that is the question we asked. So how does task-related refixation behavior affect Saccade planning, which means for targets and distractors? And as I said, this was part of an effort to study EEG during such tasks. So we looked at the Saccade-related potential, or SRPs, which is the EEG activity in the interval preceding a saccade. And what we did was we compared first fixations and refixations for targets and distractors. Now, of course, to do this, we had to co-register EEG and eye movements. And what happens essentially in this is that you can sync EEG activity to eye movement events, then you can see how does the brain respond to, let us say, what is there at gaze, or before it, in our case, for example, because we're interested in pre-saccadic activity. Now, let us look at what happens in the brain during a saccade or around an eye movement. So here you see the saccadic spike activity, which happens at saccade onset. And here you see the lambda activity, which is the brain's response to the input at gaze. And here you have the pre-saccadic activity, which is the interval where we are planning for the next saccade, and that shift in attention to the new location, this is where it happens. So this is the area we are interested in. Now here's the problem. When you talk about EEG and eye movements, the EEG is not only affected by the information at the fovea, but it's also affected by eye movement parameters. And in an earlier study, we found that the interval that we are interested in, the pre-saccadic activity, is affected by the preceding saccade size and the preceding fixation duration. Now, if these two parameters differ systematically between our experimental conditions, then we have a problem because we have a confound. So how do we solve this? One solution is to model the eye movement parameters as predictors of the EEG. We saw in two earlier talks and earlier sessions today, one by Benedict and one by Anne, that these can be modeled non-linearly. Another approach is just to select eye movements with that match in these parameters across the experimental conditions to ensure that eye movement parameters do not differ between what you're comparing. And we chose this approach so that we will be able to compare our SRPs to traditional SRPs related to saccade planning that have been seen in literature. So now let's first just look at the eye movements. So as you see here, you have the first fixations have longer fixation durations than refixations, and this is what you would expect because perhaps they are attending longer, there is more working memory or more processing load in this, and this is all good. But this is exactly the kind of thing that can also affect EEG, and therefore we have to match these parameters, right? So this is how we did the SRP comparison. We put together all fixations and refixations, and we selected those that were matched for duration, saccade duration, and rank. Rank meaning where during the 10-second trial do these lie, so that we can pick up those epochs that are all happening at the same time points of processing during a trial. And then we selected 160 millisecond epochs before the onset of a saccade. And just remember that this is the tail end of a fixation on a target or a distractor, which means that we are trying to ask, how does the current item, which is the target or the distractor, affect subsequent saccade planning? We chose 8 regions of interest corresponding to the 1020 system of electrode placement. And so we averaged about 56 electrodes here. And then we computed average SRP amplitudes for first fixations and refixations. And this is what we found. So for target, what we found is that there were higher SRP amplitudes for first fixations and refixations in the occipital areas, and a reversal in polarity for the frontal areas. And this is very neat because this is similar to a negative parieto-occipital wave that has been found during saccade planning, which means that we seem to have found some kind of an analog under free viewing conditions for saccade planning processes. And just as we expected, there is a difference between first fixation and refixation, which may be because of the processing differences between the two. This is what happened for distractors. We did not find any difference between first fixations and refixations, meaning that there were no processing differences in between the two, meaning there was no effect on subsequent Saccade planning. Because we had pretty much decimated all our electrode data, we had a dense array net. What we did was we also supplemented this average SRP analysis with some spatiotemporal clustering, cluster-based permutation tests. So essentially what happens is that you try to identify continuous points in space or electrode space and time, and you call this a cluster, and where continuous points where there's a difference between the two conditions that you're comparing, you call this a cluster. And now these clusters are then compared against a permutation distribution to identify significant clusters. And this is what we did, both for our targets and distractors. The advantage is, as I was saying, you can use all your electrode data. And here again, we found very similar results. We found two clusters, an occipital cluster and a frontal cluster. And as you see, the amplitude of first fixations was higher than refixations SRPs, and the opposite was seen in the frontal cluster. We did not find such an effect for distractors. So to summarize, we asked this question of, does Saccade planning or preparation during fixation or refixation depend on task relevance? And the highlight was trying to do this under previewing conditions. So we were able to study the interaction between top-down and bottom-up control of eye movements in EEG under previewing conditions. And what we found was that higher SRP amplitudes for first fixations and refixations were seen only for targets, indicating a difference in Saccade planning. And this difference can be attributed to the high processing costs or working memory for first fixations, which can in turn affect Saccade planning, which means disengagement of attention from the current location and shift to the new location. And this can be expensive for first fixations. That will be all. Thank you all for listening. This paper has been published. There are more details about the complicated matching procedure that we used in the paper as well. I think Huriye has probably shared the link.
It's in the chat.
Yeah, it's in the chat. Then I am open to questions.
Thank you, Radha. It's a beautiful study. If you have any questions for Radha, please ask them in the question and answer panel, and we are happy to answer them. Do you think, while we are waiting for your questions, a quick question for you. If it seems like your task is hard, at least for the design you showed, it's very tiny images, right? And TNL is very confusing as well. Is there a specific reason you made it that hard, or?
Yes, it was because one thing we wanted was that we wanted that participants fixate on the particular target to be able to discriminate it and do not see it peripherally, so that we can have more precise classification of what is a fixation on a distractor and what is a fixation on a target. They did pretty well, though, about 75 to 80% performance, though, in spite of that, how difficult the task was. Yeah.
Mohammed Abdel Haq is saying for subjects to have fun. They
did. Yeah, they did. Actually, we gave feedback, right? So at the end of every block, we told them saying you have 80% or 90% results. And given that it's an EEG and a difficult setup, people were motivated to do better and beat themselves from the previous block. So it was a bit gamified in some sense.
Yeah, that sounds good. Thank you very much. If there is no more questions, that's the end of our sessions today. And I would like to thank